This is part one in our two-part series on transcription and translation. We will be covering various aspects of the central dogma of biology in these next few concept videos. The central dogma is really the story of how cells read their genome. DNA is the most basic unit of life, and transcription is a process that reads DNA and converts it into RNA. Translation then converts RNA into protein. However, there are several other processes, such as reverse transcription, that converts RNA back into DNA, as well as DNA and RNA replication processes. I added a little dashed arrow here to indicate the process continues after protein is made, and that is described by phenotypic changes facilitated by these proteins. So what is DNA? DNA stands for deoxyribonucleic acid, and it's akin to coding for life. Here you can see that there are two classes of nucleotides. These make up DNA. There are pyrimidines, which consist of cytosine and thymine, and there are purines, which consist of guanine and adenine. These follow Watson and Crick base pairing, where you see adenine paired with thymine and cytosine pairing with guanine. Guanine and cytosine are held together by 3 hydrogen bonds, while thymine is held to adenine with 2 hydrogen bonds. Each of these bases is bound to a ribose sugar phosphate group, and this will end up creating the backbone for DNA helixes. Transcription is facilitated and carried out by proteins called RNA polymerases. There are 3 different polymerases in humans, and we call them RNA pol 1, 2, and 3. Each of them is responsible for transcribing different RNAs. So as I mentioned, there are several different types of RNA that can be made in a cell. Thus, there are several different types of polymerases that facilitate the transcription of these things. RNA pol 1 transcribes most ribosomal or rRNAs. RNA pol 2 synthesizes messenger RNA and some other small RNAs. And RNA pol 3 creates transfer RNAs, 5' srRNAs, and other small regulatory RNAs. In order for RNA pol to accurately be able to transcribe a gene into RNA, the machinery has to have a way to recognize, all right, this is where I start. So there are transcription start and stop sequences that dictate where RNA pol begins and ends transcription. The start sequence is really called the promoter region. This region includes the TATA box, which is a sequence of TA, 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 about 30 base pairs upstream of the transcription start site. This promoter detection begins with the binding of general transcription factor TF2D binding a TATA box via detection of the TBP, or the TATA binding protein subunit. Binding causes significant distortion of the DNA and kind of acts like a big flag that there is an active promoter site region in a sea full of genes. Then TF2B and TF2A bind. TF2B helps position the large RNA pol at the exact place where it should be on the promoter site. Then RNA pol 2, TF2E, TF2H all bind the DNA at that site. This forms the transcription initiation complex now. TF2H includes a helicase, which helps separate DNA by hydrolyzing ATP to expose the template strand of DNA. RNA pol then begins transcribing small bits of DNA at a time in a stop-and-go process until it can clear the promoter. Phosphorylation of the RNA pol's C-terminal domain, or CTD, by TF2H is essential for the next steps of the process. When the C-terminal domain is phosphorylated, some capping proteins will be recruited, and their job is to stabilize and cap the 5' end of the nascent RNA strand. Once they cap with guanosine and they methylate, they will be removed from the C-terminal domain, and splicing proteins will hop on and begin splicing the introns from the RNA that's being made. After they have completed their job, 3' capping proteins will bind and do their job. So what else is needed for transcription to really take off into the elongation phase? Well, the cell has to say, we want this made, but there's nothing specific about the machinery that we've talked about so far. Well, chromatin modification proteins, various transcription factors, mediators, cofactors, all of this allows specific bits of DNA to be accessed and transcribed by this machinery. So let's get into how this really happens. RNA pol binds the DNA and synthesizes RNA in the 5' to 3' direction off the template or antisense strand of DNA. Once a transcript of RNA is made that's about 10 nucleotides long and is stable, RNA pol clears the promoter region and enters the elongation phase of transcription. This follows Watson and Crick base pairing, where nucleotides added to the 3' hydroxyl group of the previous base via nucleophilic tack on the incoming phosphate group make a large strand of RNA. The difference between DNA pol and RNA pol here is that RNA pol incorporates uracil instead of thymine. The byproduct here will be pre-mRNA. Once the pre-mRNA is made, it's immediately modified. Remember that the C-terminal domain of RNA pol facilitates the recruitment of proteins that will add a 5' guanosine cap, will perform 3' cleaving, and facilitate the addition of a 3' poly-A tail. The purpose of this tail is to prevent against exonuclease degradation at the opened hydroxyl end, and also helps facilitate and stabilize more efficient translation in the future. Splicing also takes place via the spliceosome, which we will cover in our next video. After these modifications, the mRNA is ready for translation into a polypeptide or a protein.